But up first tonight, we look at the latest in medical advancements by focusing on one of the most common of all genetic problems. Sickle cell disease affects 1 in every 375 African Americans, as well as those with Mediterranean, Caribbean, Arab, and East Indian ancestry. But as Dr. Tom Linden reports, with new advances in genetic research, scientists here in North Carolina hope that they may find a solution to the problem.
Meet 13-year-old twins Vanessa and Valerie Sumner, their 15-year-old brother Victor, and their mother Gloria Amarte. They all have sickle cell disease.
Sickle cell is very painful and it slows you down 2 years late, and you can't do regular stuff that other kids do.
The disease is genetically determined. That means Vanessa, Valerie, and Victor inherited sickle cell disease from their parents. Gloria's from West Africa, where sickle cell evolved as protection against malaria. In the U.S., about 1 in 800 African Americans has sickle cell disease. You can think of a normal red blood cell as being a lot like a Cheerio. The problem in sickle cell anemia is that the cell folds up like a sickle, and that can lead to blockages in blood vessels, even leading to a stroke. That's what happened to Victor.
We were in the car one day coming from home, from the mall, and he just went out. I thought he was joking with me.
When we got home, he said he couldn't move or get out of the car, so we had to grab his arms and legs and pull him into the house.
Here they come dragging Victor in. I said, what's the matter? They said, Victor can't walk, he can't even do anything. Then I have to call 911.
Luckily, Victor recovered from this and a second minor stroke. Dr. Rupa Redding-Lollinger says he responded well to treatment, but life will never be the same. Like all sickle cell patients, Victor can expect to have bouts of fatigue and pain.
Fine. Squeeze my fingers. Tight as you can. Oh, that always hurts. Okay. Okay, good.
Once a child with sickle cell disease has had a stroke, then they need monthly blood transfusions to prevent the recurrence of stroke, another stroke, which could leave them severely damaged or could even kill them.
If, and this is a big if, Victor's body could be told how to make normal and not sickled blood cells, transfusions would be unnecessary, and the complications of sickle cell disease might decrease or disappear. The key to this theoretical treatment is finding a way to help his body make the correct form of the protein that carries oxygen in red blood cells. That protein is normal hemoglobin.
The genetic research which may affect sickle cell disease patients in the future, which we are most excited about, is the possibility that a normal gene, a gene that codes for normal hemoglobin, could be inserted into the cells so that a person who was born with sickle cell disease would be given the ability to make normal hemoglobin.
Dr. Jean-Michel Voss at the University of North Carolina at Chapel Hill is working on just that. His research is part of the Human Genome Project, one of the most ambitious endeavors in science history. The project is an attempt to identify the code that helps determine how we're made, our genome. The first step is to identify all our genes. Scientists have decoded or sequenced 1/3 of them, and that's ahead of schedule.
The original goal was the year 2005 to sequence the whole human genome. It'll be done before that. It's clear that now it'll be done probably around year 2002, 2003.
A more complete understanding of the human gene structure will undoubtedly yield startling answers, but also lead to profound questions.
Now we're moving into an area where we'll be treating people not with drugs, but with genetic material itself. That's a very different way of thinking.
And it's an area rife with ethical controversy. Researchers must be cautious of overselling the immediate benefits of their work to patients.
One of the reasons it's terribly important is that people involved in gene transfer research or gene marker research often enter this with the expectation that there will be therapeutic benefits for them, when in fact, more often, one is serving future generations by hoping to find some kind of remedy.
To that end, at UNC-Chapel Hill and elsewhere, scientists are working on ways to insert genes into cells. The theory is that scientists can then use this approach to make abnormal cells healthy. Take these cells, for example. The yellow dots are genetic material that researcher Voss has successfully inserted into laboratory test cells. The UNC scientist has also put genes into mice. These genes code for normal hemoglobin, but it's a big jump from treating mice to treating people. For patients with sickle cell disease, there's no easy remedy or genetic quick fix. But researchers are working hard to find new treatments.
Well, basically, it's finding cure for diseases, human diseases, and all you have to do is walk across the street and walk into the main hall of the hospital and seeing all these patients affected by different diseases. It would be nice to find ways of curing this in a more effective way.
Despite their optimism, researchers recognize the great difficulty in finding a genetic treatment for sickle cell disease. And even if a genetic therapy is found, patients and doctors will still have to decide about the ethics of genetic manipulation.